Thank you. Thanks, Malcolm. It's great to be here. My dad's family is from Michigan, and my grandparents' alma mater is Michigan State, so it's great to be here. I think I was here once as a child for a wedding or something, so my only memory is like a little chapel by a river. This was very lovely. So it's great to be here, and I'm excited to talk about sort of a synthesis of work I've worked on in the past, and also really new stuff. So this will be a fun sort of new talk for me, and hopefully it'll be interesting for you guys as well. So this sort of captures a lot of what I'm going to be talking about today, really looking at how we can use large-scale spatial predictors like Landsat satellite image data to predict something that's happening at a fine scale like wood growth within a tree. And we're going to be looking at some different disturbances that are related to that. And this image, just to kind of get you started, the forest on this hillside is actually being defoliated. This is like August of 2016 in a part of Pennsylvania that was defoliated that year. So sort of the lighter areas of trees you can see there are actually somewhat disturbed, but this wouldn't jump out at you maybe from an aerial photograph or maybe from a satellite image in ways you typically look at them. So a theme that's going to come up today as I talk is partial canopy disturbances. So these are changes to the forest canopy that can have effects on wood growth, but they're not stand replacing disturbances, which we understand in sort of forestry extent, and they're not always as easy to detect from something like a satellite sensor. So this is that same area. We can see this. You would walk into this forest, you might think this is spring. This is like when the little leaves are coming out in May. This is actually August because these trees have all been defoliated by a hardwood defoliator, and then they've tried to regrow some of their leaves. So there's a lot of light coming through. There's a lot of changes to this forest environment that can have impacts on the forest. So defoliation is a type of disturbance that causes this partial canopy disturbance, and that's something that I've studied a lot and I'm going to talk about as a recurring theme in this seminar today. So just thinking more broadly about different types of canopy disturbance, there are sort of hardwood defoliators, but there's also defoliators of conifers. We can see some examples of that here. The Smoky Mountains, I believe actually this was a beetle, that was probably Balsam woolly adelgid damage there. But you can see that there's sort of irregular disturbances that are affecting things. And we have some different defoliators here. You can see sign of gypsy moth cocoons and egg masses when their population's high, some other larvae, and some defoliated conifers in a different part of the country. Some of the measurement methods I'm going to be talking about are the sort of build up what the type of research I do revolve around measuring things in the field, sort of physical measurements that we can take over time, and then also measuring things from space or from a remote location to get a larger landscape scale understanding of what might be happening. So of course we're hopefully all familiar with things like tree rings, where trees leave this annual record of growth every year, so you can get an idea of, if you sample those and measure those, you can get an idea of whether growth was good in that year, so conditions were good for that tree or that forest, or whether it was poor, and then you can try to understand what caused good or bad growth. So a lot of times we look at climate or weather, what was the weather like in that year that caused the wood to change in that way. So we take cores out of trees. You can see people doing that, well, maybe not any of these pictures, but we do sample things in that way, and then we try and relate them to things to create maps of things like defoliation in different areas where it's occurring. So that's the other theme that will come up a lot, is using tree rings along with satellite data to understand tree growth and wood growth at a larger forest scale. So continuing sort of these tools and methods, this is a picture of the Landsat Sensor. Hopefully most of you are familiar with it. It's sort of one of the longest running sensors of multispectral data that we have. For the TM sensor, which is the most commonly one used now, it kind of extends from the mid-'80s to the present day, and I'm sure people are working on continuing that record going forward. So that gives us a nice large-scale view from above of what might be happening with forest canopy. Some other tools that are becoming more common, working with Google Earth, and now Google Earth Engine, which is a cloud computing platform which lets you access a lot of these data remotely, and I'll sort of touch on that because it sort of made a lot of things possible that didn't used to be. But this is increasing our ability to access data that used to be very cumbersome to manage in the past. And again, all this data needs to be linked up usually to really make sense of it. So other types of data we collect is measuring sort of plots, so measuring an area on the forest. This is something foresters have been doing for over 100 years to understand what growth is like. So things we measure in those plots are things like height. What is the diameter breast height of this tree? What species is it? How many of that species are there? But then we also look at things like how many small trees are there? What's the regeneration? So usually we have a lot of different types of data to understand the dynamics in the forest plots we're working in. But there's some unique things about the tree ring data that we work with that I'm going to go into in more detail because it's not sort of the traditional way of collecting tree ring data and makes answering some of these questions a little more possible. So a lot of my postdoctoral research has focused on working with large tree ring data sets that were collected in different parts of the country, but I focused mostly on this data set in the Superior National Forest in northeastern Minnesota. So these were collected in 2010, and they were part of sort of a large-scale forest monitoring program for the national forest there. And so across the forest, you can sort of see the boundaries of the forest here in northeastern Minnesota. We went to 36 areas in the forest, and then within those sampled 3 plots each. So we have a total of about 108 plots that were measured in different forest types that are representative of the main dominant forest types of the region. And the unique thing about this type of data that's becoming a little more common is that we took tree cores from all the trees. So usually traditional dendrochronology goes in and is trying to sample sort of the most sensitive trees to get an idea of what's been happening in climate over a long time. Like maybe you want to reconstruct the historical climate for a region. But in this case, we want to know what is the whole forest doing and how can we best understand that. So every tree that was over 10 centimeters in diameter was cored and measured, and all the tree rings in the cores were then measured as well and cross-dated. So for this research, we're interested in how does wood growth vary with annual climate. So yes, we wanted to understand sort of what the climate response of different species is, but we were kind of interested, if we look at the ranges of the main species in this study area, the blue kind of are overlapping species ranges as estimated by Little in 1970s, I think, for a lot of northern species that have sort of the center of the range in Canada. So that's things like trembling aspen, jack pine, paper birch, and things like that. But then we also, being in northern Minnesota, as you are here, we're sort of in this transition zone. So you're at an edge between the boreal forest in the north and the temperate forest more in the south. So you have a lot of these species whose distribution is more southerly historically. So we were interested in sort of looking at, well, what does it mean if these species are growing at their southern edge, where they're more exposed to warmer temperatures than the mean in their range? Or what about the species that are growing at their northern edge? So they might be, as the temperature warms, they might be exposed to like more favorable conditions than they've known in the past. And we sort of looked at these sort of species varying relationships. And our expectation was that some species sort of at their warm edge would be more sensitive to things like temperature, like drought, whereas species at their cool range edge might be more responsive to things like temperature. So one difference about this data set, again, was that we had all these trees cored, so we could look at an aggregate of tree growth. The other approach we did was that we estimated biomass from our tree ring data. So we reconstructed the diameters in every year using that measurement that we have for each year. Then we use allometric equations to estimate what the biomass would be. And this is just a little bit different from traditional methods. Traditionally, you look at ring width, so that's just the radial growth, which is a linear measurement. And you see something like this. It kind of looks like it declines as the tree gets bigger or over time. Or you standardize it, so you can just highlight what the residual is that relates to climate. So these are the same trees, actually. This is a small ash tree and a big oak tree. So they look kind of similar if you look at them in these two variables. But if you convert them to biomass increments, so this is a very large statured tree. It's tall, so all that wood has to get laid over the entire stem and all the branches. It's a huge difference in biomass versus what this little ash is doing. So even though the measurements are sort of the same in that linear measurement, there can be these really different dynamics going on. And when you're interested in like carbon budgets and things like that, you really have to pay attention to that because the conclusion, just the inference you make from the data can be different if you're not looking at that. So that was another thing we wanted to focus on. So we took all these tree ring measurements and sort of worked to scale them up to the stand level. You can see here the trees are numbered because this is a monitoring program, so we can look at this black spruce, number 42. It has a biomass time series from the tree ring data. So you can see it kind of got established in the 60s and sort of piddled along for a while this neighboring jack pine sort of added on a lot more biomass. It was sort of a better competitor. And then we can look at one more tree here. Another jack pine had a different pattern of biomass growth over time. Here we have a stem map of where all these trees are located. But then you can aggregate this up and get a signal of what is the aggregate growth over time for this forest and this plot. And I'll come back to this, sort of thinking about how do we scale up to work with Landsat data or any kind of remote sensing data. You're dealing with a pixel, so sort of a square or a measurement on the ground that's measuring multiple forest canopies at once. So you have a sub-map and the satellite or remote sensor is observing something like that. So it's going to be sort of measuring something aggregate, which is what we're getting at when we add all these tree rings together. So we modeled tree growth based on a number of potential drivers that we thought would affect growth. And this was actually work with Andy Finley, who's not here today, and some other colleagues. But we really wanted to understand sort of what is the relative importance of when you measure these things. Like how does tree age and size affect how much biomass it's putting on every year versus those differences in annual climate, like temperature and moisture stress, and some measure of stand competition. So these models allowed individual responses to vary and also allowed species responses to vary. So we could look at if some species or some individual trees were responding to different climate variables uniquely. And what we found was, these are sort of the parameters from the models that we fit. What we found was if we looked at the effect of DBH on what annual biomass growth was and age, these were very large effects on growth. This is a standardized coefficient, so these are all on the same scale. So you had a strong positive effect on growth for a very big tree. And there was a slight correcting, like a negative effect of age. And these are sort of interacting properties. So basically, if you have age, that's additional information that helps you know how long it's taken the tree to get to that point. So if it's a big tree and it's very old, then it's grown a little smaller than a big tree that's younger. But our climate variables, they did have significant effects, but they were sort of much smaller relative to these effects. So one of the things that you can conclude from this is just that we need to keep working to map and model what the structure of our forest is right now, because we don't actually have the ability to map the forest age structure, the forest size structure completely accurately today, but that's one of the biggest factors driving how much biomass sort of the forest in aggregate is laying on in any year. In terms of how individual species responded, these are sort of two, let's see, these are codes for species, so the first two letters of the genus, the first two letters of the species names, and they're arranged from northern range species to southern range species. So Picea mariana is at the top, and we have red maple at the bottom as the more southerly species. And the beige color just means it had a significant response to, in this case, temperature and a moisture stress variable, precipitation divided by potential evapotranspiration. And the key thing just to highlight here is just that we saw some unexpected things like this. Sugar maple and red oak and big tooth aspen were all having this response to climate and moisture stress that we didn't really expect, because they're at the northern edge of their range. We thought they would be getting more favorable climate, but they were actually responding in a way that would mean their growth in the future might be more variable than it is today. So we saw some counterintuitive effects there. But overall, what we took away from that part of this research was that there is the potential for resilience of some of these northern forests to climate change, because it appears that there's a lot of individual variation in response. So if every tree is sort of responding to a climate variable in a different way, that buffers the overall response potentially to some of the changes we're seeing. So we have to consider that variability, is what I would say. But there's still sort of at least 30% of the variance in annual growth is not explained by the models like these. So there's sort of this remaining question. Well, what else is driving growth? What controls growth? And how can we model it better? So that's where Malcolm Eider stepped in. This is a grad student in your department who's been a great host today. And he took off with this same data set and said, well, are these climate responses in these trees and forests stable over time, or are they actually dynamic? So this is a figure from his paper that was out this year in Ecological Applications. And on the x-axis, we have time, and on the y-axis, we have a response variable. So this is like a standardized coefficient for a regression, would be the simplest way to think about it. And it's allowed to vary over time. So what you see is, well, if you see movement in this, that means, and the, let's see, what are those called? Confidence intervals are sort of not overlapping, then you are having this significant variation over time. So this is a really interesting paper. He looked at some different climate variables like spring water deficit and also a fall deficit and some snow variables that we didn't consider. And interestingly, one of the things that he found was that there was this interaction with disturbances. So in years where we knew there was a forest tent caterpillar outbreak, so there was a defoliation partial canopy disturbance from other research, you saw sort of a different response. Maybe it's the opposite. So in this year, these trees were responding negatively to summer water deficit from the prior year. But then if you move to when there's a forest tent caterpillar outbreak, the response changes. So suddenly there's this question, okay, well, what does this mean? And that was one of the questions raised by this research. And you should talk to Malcolm for more details, because hopefully I represented that fairly. But so this brings me to the other focus, and that is sort of this defoliation partial canopy disturbance question, like returning to, okay, so we know we can predict tree growth pretty well in tree rings. We can use climate. We can use other structural characteristics of the forest. But there's still these unknown changes in the canopy that we can't measure, and we don't know that well. We don't have an independent measure of it, right? So in the past, I've worked on defoliation research. I did my PhD at the University of Wisconsin, and I focused on trying to understand gypsy moth outbreaks in the mid-Atlantic. So this is in the Appalachian Mountains, mixed deciduous forest. And we really wanted to understand forest ecosystem responses to defoliation. And I kind of like that this sort of summarizes my PhD in one little slide, and that I never knew I had that. And so one aspect was mapping defoliation with Landsat. So can we accurately map these disturbances in a way that is more informative than traditional methods of sketch mapping and things like that that have certain limitations. Then developing a defoliation model and applying that to simulation models that allow you to look at scenarios over time. So that was the focus of my dissertation. And that was in Green Ridge State Forest, Maryland, which is this dark gray forest here. So really interesting, diverse, deciduous forest. Gypsy moth is a introduced defoliator that, gosh, do you guys have gypsy moth outbreaks in Michigan that are pretty bad yet, or? Yeah, okay. Like so when I was in Minnesota, in Wisconsin, we kept waiting. I kept waiting to see. It's basically been moving across the U.S. since it was introduced in like the late 1800s in Boston. So you kind of, when you study these things, you're interested in, is that going to happen? And I feel like there hadn't been a really big outbreak yet in Minnesota where we were working. But in Maryland, it's been there since the 80s. So we have sort of this short-ish record, but there's a record of these outbreaks. And they eat like 300 species of leaves, but they love oaks, and this is like a mixed oak forest. So this is a super happy buffet for them. But one of our focuses here was looking at how can we, if we have defoliation, you have trees with full canopy, and over defoliation, basically get this huge population explosion of the larva, which is a caterpillar here, and they're going to go through and eat all the leaves out of the trees, focusing on the host they like the best. But for this, it's a general species, so it's going to hit most of the species in this landscape. And we have some papers focused on our efforts to map that disturbance. But what came out of that was we've developed a method, actually empirical models, to map defoliation based on relationships between changes in satellite reflectance, so how that reflectance changed from a year that didn't have defoliation to a year that did in the Landsat data. So this is one of the maps produced for that. This is Green Ridge in the yellow, and defoliation is scaled from green to red. 100% defoliation. And I apologize if you're colorblind. My husband's colorblind, and I should improve these maps from back in the day. So we can see patches of defoliation here from an outbreak that happened in 2000, and this continued to 2001. And the goal here was to have a continuous measure, so really capture that gradient of canopy disturbance so that you're not just looking at like, this area was disturbed, this was not, but I don't actually know what happened to those trees in any quantitative way. So we had changes of canopy reflectance, and we were actually measuring defoliation on the ground by collecting. You put out these plastic traps, and we were collecting caterpillar poop throughout the season when this outbreak was happening. So we had coincident data. It's physical measure that we can then reconstruct what the canopy biomass of foliage that was eaten was. So that was sort of the approach we used, and we also took indirect measurements like estimates from visual estimates, basically, which has actually worked pretty well when compared to this data. So here's just another example. This is Landsat data. You can see this gray area was one of the defoliated areas in this landscape, but in 1984. So that was one of the first outbreaks of gypsy moth in this landscape. The red area is foliage. It's basically like a near IR type image. And you can see we had pretty strong relationship between predicted defoliation removed. So this is sort of a defoliation metric from that paper and what was observed. And this was across multiple years of data, so this was sort of a generalizable model that we could use to map defoliation from Landsat data in new areas as well. Okay, so this is sort of a side note, but one of the other things that came out of this research was a review of all of this insect defoliation impact literature that is very intense, and but not always easy to compare. So you have all these impact studies where people go out and they say, well, I measured this much defoliation, and I measured this much change in wood growth for these different defoliator species. But they almost always have different metrics for what is defoliation, and I might say, oh, 40% is not defoliated, and then 60 to 100% is heavily defoliated, but everyone has a different metric. So for this review, what I did was work really hard to take all the data that people had published that had both defoliation measured in the field and a response, like changes in wood growth or mortality, and then put them on a scale that could be compared across studies. So this included over 37 field-based studies, 24 tree species, and 11 defoliators, and looked at tree growth and mortality. And so these are looking at growth responses to defoliation. So if we sort of relativize growth, 100% would be sort of what you expect without defoliation. 0 would be no growth. And then we just sort of sum up annual defoliation. So every year you can have up to 100% defoliation. That's the x-axis here, cumulative defoliation. But what we see is some differences among sort of functional groups of trees. So oak species that are ring porous seem to have this relationship where they sort of had a linear response to defoliation, but it was a slow change in growth. They don't sort of stop growing completely when they're completely defoliated. Whereas a number of other species, sort of these are diffuse porous, and some conifers would decrease their growth more rapidly. So if you have 100% defoliation here, a lot of these species are sort of at 20 to 40% of growth under 100% defoliation, and then they're going to level off because you can't have negative growth. And then the conifers, which are storing so much of their energy and their reserves in their leaves, if they're defoliated, what we saw is most of them are shutting down growth completely between 100 and 150%. So up to 2 years of defoliation, if you think of it. So this is just to show we have these now sort of theoretical relationships that are based on empirical data that we can go and look. If we could map defoliation and we could see the wood growth response in tree rings, would it look like this? Would it look like all these other types of data that people have measured? And can we start then to think more about how forests are responding in a way that might feed into models at a larger scale? Okay, so thank you for being so patient with me on this talk. This is a lot of stuff. So working in this Minnesota tree ring stuff, for a long time my goal was like, okay, we have to take this defoliation model and map defoliation in this area and predict tree growth, because we have this awesome tree ring data set and we know how to do this. These models work pretty well. So this is northeastern Minnesota again, where our tree ring data is located. And so I started doing this, and I think Emily we were in the same lab for a while, and she saw me working on this a long time ago. So this is sort of a defoliation map from 2001. In this case, the lighter colors are 100% defoliated. Dark green is not defoliated, so it's happy forest canopy. So we can see here this is the Bayfield Peninsula and the Apostle Islands. There was a lot of defoliation there in 2001, but also some areas here on the northeastern shore and up near the boundary waters. And if we zoom into where one of our plot locations is, we have 3 plots in this area. In this case, they're just labeled P1 through P3. But you can see a couple different defoliation outbreaks here. So this is 1988, 89, and 90. The lighter colors are defoliated. So in 89 and 1990, we sort of see a little bit of defoliation in this site. And then if we jump ahead, 98 through 2002, this bright color means like really defoliated, because 100% is the site. So it was almost 100% defoliated at this site, 2001. So this is getting kind of closer to what my idea was a while ago, what we could do. So using this data, sort of applying that approach we developed in Maryland, and then plugging in our tree ring data that we collect in Superior National Forest, you could start to look at whether tree growth, so in this case the black line is it's relativized growth, but it's that stand level, plot level biomass increment. So it's from the tree ring data, all the trees are added up, and then it's relativized so that 1 is sort of what you expect. And the red line is the mapped defoliation disturbance from Landsat. So for this particular plot, it's an aspen plot. I think it's the P2. You can see, like, yes, in 1989 and 90, there was some kind of disturbance there, and the foliage seemed to rebound. And then in 2000, 2001, we see that defoliation signal in the Landsat data, and then we see it in the tree ring data. So we're seeing a pretty nice correspondence between this Landsat disturbance index and the wood growth from the tree rings. We can look at a completely different forest type. So this is balsam fir, and the black line again is this aggregate wood growth. And so this was a stand that was sort of established sometime in the 80s. It's a young stand of balsam fir, and so it's growing. That's just sort of your typical, wood growth curve. And we can see that the green in this case is showing the Landsat defoliation index, or the inverse of it. And it's tracking it pretty well. So we're seeing some nice relationships in some of the plots that we looked with initially. But the challenge here is, how do you scale this up? So we have sort of developed these different methods, and we're really interested in being able to map defoliation everywhere and then predict tree growth everywhere. That would be amazing. But this is our study area. Each square here is a Landsat footprint. So each square represents 1 scene of Landsat, and these are all our plots. So to get all of our plots, we'd need, like, I don't know, if this is 9 or 10 Landsat scenes, and then you would need sort of all the years. You would need a good growing season image from all those years, and you would need there not to be clouds, and there's lots of issues that make it challenging. And so we had used this anniversary date approach, so we were taking an image that didn't have defoliation and using that as a baseline to then model defoliation. But this depends on there being a good baseline, and there isn't always one. So Landsat comes every 16 days over the same point, so you may only get, I don't know, I'm trying to think how many days. You only get a certain number of observations over the growing season, and oftentimes there's clouds. We just live in a cloudy environment. So this sort of is one of the limits that's held back sort of taking this approach and scaling it up to a large scale. So now I just sort of want to talk about some new things people are doing to try and get beyond this, and sort of as an approach we've adopted. So this is some work from Valerie Pasquarella. She's a postdoc at UMass who was at Boston University as a PhD student. And what she and a lot of other folks are starting to do now is take all of the Landsat observations, so not just like that one scene from the growing season this year, that other scene from the growing season last year, but just suck in all of the data. So each dot here represents an observation from a Landsat scene, a different date. So you can see here there's a lot of observations over time. And this, was made, the Landsat archive was made free, I don't know how long ago it was, like maybe 10 years or something, but that introduced this whole opportunity if you have huge storage capacity, because these scenes take up a lot of space. But what we see here is there's a lot of data available if you can do it, and you can line them up. What she's done is line them up by day of the year. So all of these, 1985 to 2015, are lining up, and what we see, in this case, she's looking at an index that sort of gets at greenness from the satellite. You see sort of a growing season curve. So this is sort of a phenology curve that you're probably used to. Leaf off is here and sort of leafing up, and then you have growing season where the canopy is full of leaves, and then we have fall and then the winter. So you can sort of create this day of year phenology pattern that can be useful in a number of ways. And she's sort of highlighted here, just this green, the green color just means growing season images. The blue is leaf off images. So there's a number of people sort of working with this type of data to look at changes in phenology with climate change, or even nitrogen use and limitation. But one thing Valerie's done that is sort of tying into what I'm talking about today is they had this huge outbreak of Gypsy Moth in New England in the last 2 years. So she took their method. So this is kind of spreading these images out. So this is just for 1 year, 2013. Each dot is a Landsat scene. You can see here that you don't have as much data for each year as if when you look at all the years together. So you can get the best images and sort of start creating this time series of what the phenology of that plot is, but you don't always have as much data as you want. But in 2016, what they saw was this strong defoliation signal. So early spring, you start having leaf out, but then the defoliator comes, and those caterpillars start eating everything, and you never get anywhere close to your maximum leaf area during the growing season. And so the nice thing about this approach is you can actually get, instead of having an actual image that is your baseline of this is a full canopy for this area, you can get sort of a synthetic image because you have this average over time. And that can become your baseline. And so this opens up, this helps deal with a lot of the problems that we were encountering with our approach. So this is a nice map that they made. You can see here some Gypsy Moth defoliation. Most of Rhode Island was defoliated. It's the red color. And they just sort of used a residual as their metric of defoliation. And I think this worked pretty nicely. And it'll be interesting to see what the 2017 maps look like. But so what are the challenges to scaling up using that approach to our area? Like, can we transfer that and do it here? Well, so here's the thing. So this is like from the Boston University sort of Curtis Woodcock's lab. So this is a group that's worked with NASA and Landsat for a long time, and they have like these enormous servers. And you can see here, this is one Landsat footprint and another Landsat footprint. So they're just working with two. So they have all this assistance and these huge servers, and they can get all this data for these Landsat scenes. But they can only do this for like 5 scenes for their area. And they have like all the resources. I don't know many other groups that have those resources. So this is not feasible. Like if you remember, we had at least 9 just covering our plots. And I do not have huge servers that I could take all the Landsat data. So how can I do something like what is described there for our area? Because we have limits to data storage and processing time. And some of these areas, these plots could have like 2,000 Landsat dates that I would have to get the data from to do this approach. So that's where we've turned to Google Earth Engine. And this is really, it's a great opportunity, I think, because what is available on Google Earth Engine is sort of the ability to access the entire Landsat archive and work with it remotely. So you don't actually have to download all this data. If you can figure out their JavaScript front end, you can get the data for your plots, or if you can program within their system, you can then turn around and map things anywhere that they have this Landsat record. And it's the whole record. So this is sort of, I don't know, how many of you have worked with Earth Engine yet? Anyone? Some of you? Was it a good experience yet? Not yet? limited. Yeah. So there's, like, you have to learn their programming, at least now, until hopefully they make it more accessible. But what this has allowed me to do, and my collaborators to do, is start to look at these phenology time series for our tree ring plots and use those to estimate these defoliation metrics and then create models of tree growth, including defoliation, that's sort of quantitative and independent from the other variables that we have. So these are two of our plots in the Superior National Forest. This one is dominated by 90% trembling aspen. You know, we have all the data for these, so we can say that we know what they are. This is a plot that's 90% black ash. And this just highlights, we can see sort of the mean phenology, so day of year 100 to 300. We see the green up signal, and we see that growing season is sort of flattened off. And I'm using a different index of sort of canopy moisture status because that correlates best with defoliation in our prior studies. But you can see here there's a much longer growing season for something like trembling aspen than black ash. So ash are sort of well known for leafing out latest and then dropping their leaves earliest. And this is an amazing example of that, just seeing this very short abbreviated season versus the trembling aspen. So you can look at things like this, I'm sure, species composition and differences in phenology from this types of data. And that's not the focus of what we're doing, but I think it's interesting. But what we've done is taken our defoliation models that we made in the past, so when we mapped defoliation from those anniversary date images, sort of modified it and adapted it so that now we can model it using some of these new approaches, sort of a deviation from a mean phenology versus one image date as a comparison. And so now I'm calling this the foliar biomass index, and just because it actually lets you know if foliage is increasing as well. It's not just telling you defoliation. But what this plot shows is year on the x-axis, and in black, so the black line is showing us relative wood growth. So that's one of our plots. This is an aspen plot. And it's sort of over time. We can see what the wood growth is doing in the brown and black. And then in the green is this Landsat modeled defoliation, this foliar biomass index. And we can see that it tracks pretty nicely on this plot. We had this big forest tent caterpillar outbreak in 2000, 2001. We see that the foliage dropped down to almost zero, and wood growth dropped down to, I don't know, 30 or 40% as well. And we can see here sort of an illustration. These are how big our plots are. They're about 400 meters squared. Landsat pixel is about 900 meters squared. So they're not exactly the same, but you're getting enough of a, so there's some noise in the relationship, but it's still pretty good. These are just a few more plots. Actually, revisiting those plots we looked at before, we can see some differences here. If we look at a balsam fir plot, what you expect with conifers is the phenology is they don't green up that much, right? Because they always have leaves. So we sort of see that as the stand gets older, it's sort of a flat curve. Whereas with trembling aspen, you see a pretty strong phenological signal. And again, here in the red, we have this foliar biomass index versus the black is the wood growth. And we can again see good agreement in these cases between wood growth and Landsat. And so that, if we just like plot these against each other, x-axis shows that Landsat defoliation and relative growth. This is sort of what the data looks like for those two plots. So pretty nice relationship overall. So I guess before I sort of jump into the one last bit, this is sort of where we're at right now. We're working to develop models that'll let us turn around and apply these to larger areas. And Malcolm and I have been talking about that today, how we can work to kind of improve our models and apply them in new areas. But one of the great things about something like Google Earth Engine that you don't have generally with satellite data is it's portable. So to demonstrate this, I can go to a completely different place, like North Carolina, where Duke has this free air CO2 enrichment study, the FACE study. So they have these, if you're not familiar with it, they built these like enormous structures to kind of add CO2 into the atmosphere around these forests and see what the effect was. This is a huge study. You can see different plots here from that in this loblolly pine plantation. So this is a fast-growing conifer in the south. And this is one plot, the Duke Face site. And the nice thing about something like this is they actually went out and measured diameters of all the trees on their plots. They used big plots, and they did it every year. So it's not tree ring data, but they have annual remeasurement of inventory data. So we can get the same kind of biomass increment from that type of data and see, well, okay, how does it compare to our Landsat foliar biomass index? And what we see is, this is the green line is the Landsat foliar biomass index. And we can see that this loblolly pine plantation was planted somewhere like 1985. And, or 1983 it was planted. I guess I've consolidated this information at different points. So you sort of see this, greening up pattern from a new stand, a new plantation. It kind of reaches maximum leaf area index, and it's going to be sort of averaging around this area. But what we can see is, let's see, I marked a few things on here. I guess I didn't mark when they started adding CO2 to this one. But in brown and black, we can see the stem biomass as it was measured using those annual inventory, that data. So we see sort of this pattern over time. And we can see that it's actually tracking pretty well, this foliar biomass index. We've sort of been developing more intensively in Minnesota, more in deciduous plots. So this was really exciting. They actually harvested this site in 2011. You can see that there. And there's a couple other things that jump out. They had, I can't remember, there's either an ice storm or a hurricane that corresponded to some of these big drops in the wood growth and the canopy. So there's a huge potential here. And I have a lot of other examples like this that we're sort of analyzing to understand better. So this is sort of all over all the face sites. There's sort of a relationship you expect as defoliation increases, relative growth decreases in loblolly pine. And so our ongoing work is that we're trying to develop models that predict those changes in wood growth from time series of Landsat and other variables. So we're going to add in this component to sort of the types of models we've worked on in the past, but hopefully gain really new insight and just understanding, because now we can sort of separate out. Well, we thought that was the effect of climate, but in that year it was actually this insect outbreak that was happening. And now, in some cases, that climate variable might not seem as important, or it might seem more important. And so all those things, I think, are really exciting and interesting. And that's something Malcolm and I will be working on with others on this paper that's in progress. So I just want to acknowledge a lot of the people that are also working on aspects of this project, my co-author Tony D'Amato, and a lot of other great people who are working on long-term plot data or are sort of testing these approaches out with their data to see, oh, does it work here, and what can we do with this going forward? And so that's all I have. I'm closing on this image because I was running this code overnight last week on a Landsat of New England, and when I came back, it was not a map of forest composition, but some of the data, when I displayed it, looked like this, and I thought it was really beautiful. So this is a mistake that I thought would be maybe help people think of questions. But so that's all I have. Thanks for sticking with me, because I crammed like as much as I could in there.